Hello everyone. Inshallah, in today's session, we'll be going over one of the most important lessons in Unit 1, Chapter 1b. And this lesson is called Oxygen Hemoglobin Dissociation Curve. In this session, we'll be covering number 1, the hemoglobin structure and function. We're not going to go over the details of protein structure because it's going to be a dedicated lesson, and it's an extremely important lesson that will be keep asked about, that will be keep getting questions about starting from Unit 1 till 6. So we'll go over the hemoglobin structure and function in relation to our discussion. We'll be go over key terms related to this lesson. What do you mean by affinity? What do you mean by loading? And then we'll go over the main discussion for today, the oxygen hemoglobin dissociation curve. What do you mean by the curve, the readings on the curve, how this curve can be affected. And we'll discuss how some living organisms, they have certain adaptation to their environment. So they want to make sure that they're getting the maximum amount of oxygen that they can get according to this environment. And then we discuss the Bohr effect, which is basically the shifting of this graph according to the effect of carbon dioxide. And then we'll go over topic questions. Of course, whenever we're going to cover anything in this course, number 1, the content itself will be related to the past papers. Number 2, within every single discussion, I will tell you about the topic questions. Now, please pay attention, especially if you are taking this exam in January. This topic is heavily asked in January, starting from January 2020 till January 2024. Every single past paper of these did have this lesson in the questions. And the style of questions, they vary from 3 marks up to 6 marks. Of course, the 6-mark questions, they involve multiple lessons as well. And already know about level 1, level 2, and level 3. We'll get into that into another discussion. However, they can ask you about different questions, like starting from 2 markers, 3 markers, math questions related to this. The graph questions, which is the most common question, is going to be around 4 marks. So, and it's very straightforward questions, if you understand the lesson, of course. Now, inshallah, we'll go over, as I said, we start with the hemoglobin structure and function. If I would like to start talking about oxygen, you already know from IGCSE content that we need oxygen for the cells, right? So if I'm going to make like a human in here, I'm going to make the lungs. Here. Then, of course, we have the heart. Near the pulmonary arteries. By the way, the discussion of the heart is going to be the first lesson, the first and second lesson of topic 1b, and we will discuss the lungs in relation to the surface area in inshallah 2a. And then we have the peripheral cells or the body cells. Now, why we need to learn about hemoglobin and the oxygen hemoglobin dissociation curve? Because we know that in our circulatory system or the cardiovascular system, it contains of 3 major things. Number 1, a pump, which is the heart. Number 2, a major blood vessels that are going to bring oxygenated blood into the tissues, and these are called arteries. And then number 3 is going to be the veins. And of course, the interconnection or the connection between arteries and veins is called capillaries. Now, why we need to bring oxygenated blood into the cells? So oxygen is extremely important to be pumped by the circulation, because oxygen is going to be used by the cells in something called aerobic respiration. And aerobic respiration is extremely important to get what? To get energy. So basically, without oxygen, we cannot go into something called aerobic respiration. And this process, the 4 stages, you'll discuss this heavily in the first lesson of Unit 5. But according to our discussion, we just need to elaborate about the importance of having oxygen that will be involved in aerobic respiration. And of course, you know that the energy is going to be used in multiple things. It's going to be used in all metabolic reactions. It's going to be used in muscle contraction, nerve conduction. So if you think about it, from where we can get the oxygen? From breathing, right? So we get oxygen from air. You already know that the air contains multiple gases. We have oxygen, carbon dioxide, nitrogen. So we get oxygen from breathing, and oxygen will get into the lungs, right? And you know that the of course, the structure that I'm about to draw, it's a microscopic structure, but this is just an illustration for it. Let's draw the alveoli. And inshallah, chapter 1, 2a, we will discuss the importance of alveoli, why they have this certain shape. Now, let me tell you about a hint for future lessons. Whenever you see something curved, it means that to increase the surface area. Instead of having it flat, I'm having it curved to increase the surface area. And depending on the location, we will say increase the surface area for what? For example, here, increasing the surface area for gas exchange. In the intestines, the villi, they increase the surface area for absorption. So whenever you are seeing something that is curved, in the inner mitochondrial membrane, it's the cristae, it's folded, right? To increase the surface area for the electron transport chain. So here we are having a high surface area for gas exchange. Now let's go back to our discussion. As I was saying, the whole discussion for this and why we need to go over the structure of hemoglobin, the oxygen hemoglobin dissociation curve, is that we need to bring oxygen into the cells. Now, oxygen, and this is simply by the law of diffusion, will go from high concentration gradient, right? And high pressure to low pressure here, into a lower concentration gradient, comparing the atmosphere to the lungs. And then when the deoxygenated blood comes through something called the pulmonary arteries, will come into the lungs. The structure that will carry the hemoglobin is called the red blood cells, or the erythrocytes. And by the way, you don't need to know the name erythrocytes. Yes, it's mentioned in the book, but that's fine. You can say red blood cells. Whenever they go in the deoxygenated circulation, whenever they go to the lung, they carry no hemoglobin or low concentration of hemoglobin. Now, in order for the cells to get oxygen, we need to load these RBCs. Now, you might ask yourself a question. Of course, if you follow the law of diffusion, so it's going to be from high concentration gradient to low concentration gradient. But then this same red blood cell, when it comes to the tissue, is going to unload this oxygen. So this will be the main core for our discussion. Why in here we're having a high affinity, the hemoglobin for the oxygen, and why in here we're having low affinity. Now, if you go over the hemoglobin structure, the hemoglobin, it's a protein, and it's a quaternary structure of protein. We will get into the discussion of proteins in details in the first chapter. Of course, in the specification, you will see it as part of the second topic. But I would like to discuss it from the first topic and then repeat it once we get into enzymes, because it's a very high yield concept. So it consists of 2 polypeptides, 2 alpha and 2 beta, and it contains iron as a prosthetic group. Now, every 1 hemoglobin molecule can carry 4 oxygens. So 1 hemoglobin maximum can carry only 4 oxygens. And this binding, it's called the cooperative binding concept. What do I mean by this? If I'm going to imagine that this is 1 hemoglobin, as I said, we have 4 sites, right? And the hemoglobin, by the way, exists in this red blood cell. This is a superior view from the above. So you know that it's biconcave, it doesn't have a nucleus, again, to increase the surface area. So it can carry as much hemoglobin as it can, and again, to transport oxygen. When hemoglobin is inside of the red blood cells, like this. Now, you might think that once the hemoglobin or the red blood cells are entering the lung, and when they will come into contact with the alveoli, immediately oxygen, all of the sites will be filled. And this is wrong. Usually what will happen that it's called the cooperative binding. And I will explain this on the graph. It's not a linear graph. It's called sigmoid graph. And why it's sigmoid? Because of this phenomenon. Usually the first oxygen will find it difficult to bind to the hemoglobin. But once the first oxygen is going to bind, it will cause something called a conformational change, a change in the tertiary structure of the protein. And what will happen that as oxygen is binding, it's easier for the following oxygen to bind. So this one's going to be way easier for the other oxygens to bind. And that's why in the beginning, you will see a resistance in the graph. You will see a slow rise in the graph. And then eventually, as it's going to be filling, the hemoglobin is filled or loaded with oxygen, you'll see that the graph is increasing and eventually it's going to flattens out. And why it's going to flatten? because all the sites are now loaded with oxygen. So let me recap what we did so far. We talked about the importance of oxygen. You're not going to get any of these questions, but it's very crucial and important to understand it because these are the basics starting from Unit 1 until Unit 5. So the importance of oxygen, of course, is going to be used in a process called aerobic respiration. What is respiration? It's the release of energy within the cells. We do have two types depending on the presence of oxygen. If we have oxygen, then we call it oxidative phosphorylation or aerobic respiration. And if we don't have oxygen, it's called anaerobic respiration. And we already know that anaerobic respiration provides us with little amount of energy compared to aerobic, and we have lactic acid as another product. Now, so in order for us to the cell to have this aerobic respiration to get energy in the form of the universal form of energy, it's called the ATP. We need to bring oxygen for these cells. And not only this, these cells are active cells and they do have reactions, right? So one of the waste products, not only, you know that some of the waste products will go through the kidney. Some of them will be released by the digestive system. And some of them will be released, one of them is a gas, it's called carbon dioxide. So we need to bring nutrients plus oxygen to the cells, and we need to remove waste products, and one of them is carbon dioxide. So we will talk about all of these in a graph and how it's going to be incorporated into the graph. And then we discussed the hemoglobin structure. We said it's a quaternary structure of protein. It consists 2 polypeptide, 2 alpha and 2 beta. This discussion will be very important in 2 sections. Section number 1, the amino acids and the protein structure, how it's folded, the quaternary structure, primary, secondary, tertiary, quaternary. And eventually, it will be discussed in fetal hemoglobin, because fetal hemoglobin consists of 2 alpha and 2 gamma. Now, we have a difference in structure. Let me tell you something. Whenever you hear about a difference in the structure or a certain structure, you must relate it to its function. So the fetal hemoglobin is different from the adult hemoglobin. So it contains 2 alpha and 2 beta, right? So this is the adult. The fetal is going to 2 alpha and 2 gamma. So it must be, there's some sort of difference because of this gamma chains. And then we talked about the iron as the prestator group. And we said that every hemoglobin consists of 4 subunits, 2 alpha, 2 beta. Each subunit combined only into 1 oxygen. So in total, 1 hemoglobin combined to 4 oxygens. And these sites are not loaded immediately. They are loaded one by one. And we call this cooperative binding concept. So here, we call this loaded hemoglobin. We call this unloaded hemoglobin. Usually, if you're going to think about this, where is the site in our body that contains the highest concentration of oxygen? It's going to be where oxygen comes into first place, right? It's going to be where? In our lungs. So if I'm going to think about it, it's going to be 100% saturation or loaded where? In the lungs. And where we need to drop this oxygen into? We need to drop it into the respiring cells, right? The cells that will break it down in aerobic respiration. So it's going to be what? This is going to be respiring cells. Cells undergoing aerobic respiration. So all of this, with the exception of cooperative binding and the structure of hemoglobin, the rest is just for understanding. By the way, as we are going through this course, I will tell you this is for understanding. But this, you must know this because you're going to be tested in this concept. But of course, it's just not loading you with the information to write. You must understand the topics itself. So let's start. Let me show you the structure of hemoglobin. And by the way, within this video, you will see a full PDF that includes all of the notes in details regarding this, plus topic questions. Of course, because this is a sample, I've listed majority of questions, but of course, there's more questions related to this concept. In total, I think around all of the past papers regarding this lesson, there is a total of 8 questions. So this is the structure of the hemoglobin, as you can see in here. We have here the beta 1, beta 2, alpha 1, and alpha 2. And as you can see here, we have the heme group that contain iron inside of it. Now, we said that what is the function of hemoglobin? It can transport 2 things. Number 1, oxygen. Number 2, carbon dioxide. Oxygen from the lung to the respiring tissues, and carbon dioxide from the respiring tissues back to the lung. Now, let me tell you about something. Usually, if I'm having oxygen and carbon dioxide, hemoglobin or carbon dioxide has higher affinity to bind to hemoglobin rather than oxygen. And oxygen, when it's going to bind to the hemoglobin, we call it oxyhemoglobin. However, when we have carbon dioxide, it's called carbaminohemoglobin. Is this important? No, not really. That's why whenever I'm not discussing something, you'll see it in the notes, but it's not that important thing. So we said about the oxygen binding and how it's going to be cooperative binding, and we'll elaborate on this, inshallah, later on with the graph. But I need the affinity for oxygen hemoglobin and loading and unloading concepts. Whenever we talk about affinity for the oxygen for hemoglobin, it's related to something called the partial pressure of oxygen. Let me draw this so it can be more simplified. There we go. Here we go. For example, imagine that this is a pump or some sort of tank, and then we're having a pressure measurement. Now, if this is, let's say, air, for example, we know that air contains multiple gases, right? So we have oxygen, we have carbon dioxide, we have nitrogen. When I say the partial pressure of oxygen, it means that the pressure exerted by oxygen in a mixture of gases. In this case, in the air, it's going to be the pressure exerted by oxygen and within carbon dioxide and nitrogen. If I'm going to talk about the lung, it's going to be the pressure of oxygen exerted, or the pressure exerted by the oxygen within the lungs. So the partial pressure of oxygen means that the pressure exerted by one type of gas, whether it's oxygen, it's carbon dioxide. Usually the style of question that you'll be getting is going to be only about oxygen, because this is the main core of your discussion. Pressure exerted by oxygen and a mixture of gases. And this mixture can differ depending on what we are talking about. So we will discuss this. Where do you think the partial pressure of oxygen, or the highest concentration of oxygen in our body, exists? Again, it exists where? In the lungs. And the lowest partial pressure of oxygen, where? Exists where? In the respiring tissues. This is extremely important, because the more or the higher the concentration or the higher partial pressure of oxygen is located, the higher the affinity of hemoglobin to bind to this oxygen. And vice versa. The lower the partial pressure of oxygen, the lower the affinity of hemoglobin to bind to it. This is very important. You don't want to have high affinity all the time, because you want to load the hemoglobin and you want to unload the hemoglobin where we need oxygen. So let's go back to main key terms, and then we will get into the heavy stuff, which is going to be the graph. Let's get into this. So again, as I said, the partial pressure. So you must learn about these key terms. The only one that came across past papers at a state what is meant by is going to be this. However, it's extremely important to know the rest, because these are not going to be asked about the direct definition, but these concepts will be applied into the writing questions. Now, if I'm going to talk about what is the sort of assessment, you already know, or let me explain it, that in AS or A levels, we have three sorts of assessments. We have a style of questions. It's called assessment objective 1. This is the easiest question, and it comprises about 31 to 33% of the questions. Number 2, we have assessment objective 2, A and B. And these are the sort of questions that will start from comment, explain, the 6 markers. And then we have the assessment objective 3, which is basically related to the practicals. And in case of biology and chem, it's going to be unit 3 and unit 6. Now, these questions will be asked in both AO2. Now, the partial pressure of oxygen was in one of the past papers, states what is meant by the partial pressure of oxygen, and it's the pressure exerted by one gas in a mixture of gases. Now, let's go into these. So, what do you mean by the affinity of hemoglobin to the oxygen? It's the ability of the oxygen to bind to hemoglobin. And of course, as we are having more oxygen, it's going to bind to more hemoglobin. And as we're having a higher pressure of oxygen, it's going to bind more to hemoglobin. The saturation of hemoglobin with the oxygen, when the hemoglobin is carrying the more, if all four sides are filled, then we say that the hemoglobin is saturated or loaded with oxygen. Loading or association of hemoglobin, the binding itself. Unloading, the unbinding. This happens where? In the lungs. And this happens in the respiring cells. So let me zoom out now to show you the graph. The loading and unloading of this oxygen hemoglobin dissociation curve, this graph is extremely important. And I will tell you what is the style of questions that you'll be getting through this graph. So this is the graph. Here on the x-axis, we have the partial pressure of oxygen. And of course, you know that the pressure is measured by Pascal, right? So it's called kilopascal in here. And the percentage saturation of hemoglobin with oxygen, the four sites again, within the, of course, you know that the red blood cells is filled with thousands of hemoglobin, and each hemoglobin, one hemoglobin combined to 4 oxygen. So this is just for simplification. Now, you see that here, the percentage of saturation is 100. Where the case has 100, right? This point, right? This happens where? In the lung. And then you see the lowest amount in here, and here where? We're having it in the respiring tissues. It will start to be unloaded in the respiring tissues. Or respiring cells. Specifically, what is the structure in the lung in which gas change occurred? Because you know that the respiratory system consists of multiple structures. It doesn't happen on the level of bronchi, it doesn't happen on the level of trachea. It's lower down in a structure called alveoli, and we discussed the alveoli structure in detail, how it's adapted for its function, the concept of surface area to volume ratio in topic 2a. Now, the red blood cells that are loaded with carbon dioxide and less oxygen are entering the lungs through a vein called an artery called pulmonary artery. This is not the core of our discussion. Now, as it's getting into it, do you remember when I say that the binding of the first oxygen, it will find a struggle. It is very difficult for the first oxygen to bind. But once it's going to bind, it's going to expose the sites where other oxygens can bind. And we call this conformational change. And then it will start being loaded with oxygen, let's say 25%, and then 50%, 75%, and then 100%. Why it cannot keep going up? Because it's already what? It's already saturated. All the sites are now filled with oxygen. That's why here we're having a flat line, because you can be asked about this. Why the curve is going to flatten out? Because here, all the sites are now filled with oxygen. Why it looks like this in the beginning? Because the binding of the first oxygen is difficult. Once the first oxygen, and we know that hemoglobin contains 4 subunits, right? So the binding of first oxygen is going to be difficult. Once the first oxygen will bind, we will have a conformational change in which we are going to expose the site for the other oxygens to bind. Now that's why the shape of this curve, it's not a linear curve. If it was a linear curve, it looks like this. But it doesn't happen. Why? Because in the beginning, it doesn't fill all the active sites, or it doesn't fill all the sites immediately, and it's not going to keep going up. And we call this shape, it's called an S-shaped curve or a sigmoid curve. So the shape of this curve, or this phenomena that we are having in here, what is the reason that we are getting this sigmoid shaped curve or this S-shaped curve? It's because we're having the cooperative binding of oxygen. And again, because the binding of the first oxygen will be difficult, once the oxygen will bind, it's going to cause a conformational change in the oxygen and the sites that will allow, and because we know that hemoglobin is a quaternary structure, or a 3D structure protein. It's going to expose the sites where other oxygen can bind, and then it will flatten because it's already fully saturated. All the 4 sites now are filled with oxygen. Where does this happen? In the lungs, specifically in the alveoli, where the unloading happens in the respiring tissue, or where this red blood cell is coming from the respiring tissues. So let's go over the concepts in here. We said as the cooperative binding, and so hemoglobin exhibits a cooperative binding. Now, honestly, I don't like to go over the PDFs or read them. I will explain the high yield important things, everything you need to learn. But everything that I'm saying exists in this PDF. I don't like going over the PDFs or read them. You can do the reading, but you just have the understanding first. Every single word I've said is existing in this PDF. So you didn't need to write anything down. Now let's see something. I would like to show you a set of questions before going into this. This is the mama hemoglobin, this one. This one, yep. So one rule of hemoglobin is to transport oxygen. What is this graph? It's the oxygen hemoglobin dissociation curve. Once you see the percentage of saturation with the partial pressure of oxygen, this means we are talking about this curve. The graph shows the oxygen dissociation curve of hemoglobin. State what is meant by the term partial pressure. As we said, it's the pressure exerted by one gas or by oxygen in a mixture of gases. Explain how the structure of hemoglobin causes the oxygen hemoglobin dissociation curve to be this sigmoid shape. What did we talk about? We know that, as we said, the shape of this curve is not a linear curve because we're having the cooperative binding phenomena. And this because the binding of the first oxygen is difficult to the subunits, the 4 subunits of the hemoglobin. Once the first oxygen will bind, it's going to cause a conformational change in the shape of hemoglobin. This will cause the other oxygens to bind easier, and then it will flatten out. So let's see how many marks we got. So hemoglobin consists of 4 subunits. Binding with the first oxygen molecule is difficult. Binding of other becomes easier. Due to the conformational change, we already got the full mark. As hemoglobin becomes saturated, less oxygen combined, so the curve will flatten out. Here we have 5 marks. Yes, it's a 3-mark question, and we have 5 answers. We have 5 sentences to write. So even if usually lots of the students will not think about writing this. Let me be honest with you. Also, the examiner's report, lots of students will miss writing this one, and they will miswrite this one. But you can still get the full mark. Of course, I would recommend getting all of them, but these are the concepts, the core concepts or the core discussion for why we're having this sigmoid shape. So out of 3, you got 3 out of 3. Usually not all of the students will write these 2 sentences. However, you can get that. Writing these 3 will make sure that you are getting the mark. So this is regarding the oxygen hemoglobin dissociation curve. Let me tell you about something before we get into the rest of our discussion. Now, if we get into, do you remember when I said that the cells will take oxygen for respiration, and then they will release what? As a waste product? Carbon dioxide, right? The nature of carbon dioxide is acidic. Whenever you hear something is acidic or an acidic environment, what is going to cause to the blood pH? It will cause the blood pH to decrease, right? Now, the idea about that some of the carbon dioxide will be loaded, the other will bind through carbonic anhydrase, forming hydrogen carbonate. You don't need to know all of these details. However, what's in your specification is you need to know the effect of carbon dioxide on this curve. You don't need to know how carbon dioxide is exactly transported or what is the reaction. Of course, there's so many details we can include into this, but it's not related to your specification. So what you need to know is that carbon dioxide is acidic. And if something is acidic, it's going to lower the pH, right? And if something is alkaline, it's going to increase the pH. Now, why this is extremely important, and it will have an effect on this curve. It's because of this. We know that, go back to our discussion, hemoglobin, what is the nature of hemoglobin? Is it carbohydrate, protein, or lipid? It is protein, right? And we already know that proteins can be affected by certain things. One of them is going to be the temperature and the pH of the medium. So if I'm having an acidic pH, I can denature an extreme acidity. I can what? I can denature the hemoglobin. And when I denature the hemoglobin, what will happen that it's not going to bind to the oxygen, it's not going to perform its function. So instead of going, let's go back to our original curve. Instead of being saturated fast, this would happen like this. So let's say that this is the effect of carbon dioxide. The shape of the graph, instead of being saturated in here, it will be saturated later on because you are having difficulty to fill all of the hemoglobin with oxygen. So we are taking more time to saturate the hemoglobin, right? So the shape of the graph will be shifted where? Here is shifted to the right, and here is shifted to the left. So under any circumstances where we have a low pH, which is acidic pH, by the way, it's also alkalosis, but let's not discuss into acidosis and alkalosis. You don't need to know this for your discussion. But the effect of carbon dioxide, the higher the pH, the more shifting to the right, the curve will be shifted to the right. Now, what do we call this shifting? And it was caused by which gas? It's not oxygen, it's carbon dioxide, right? We call this shifting of the graph to the right because of carbon dioxide the Bohr effect. Because we know also that carbon dioxide can bind faster to hemoglobin has higher affinity to carbon dioxide compared to oxygen. So if we're going to go in here, the lower the pH. In A curve, where the pH is the pressure, here the kilopascal is the partial pressure of carbon dioxide. So as you can see in here, in C, we have the highest partial pressure of carbon dioxide, which means that we are having almost the highest percentage or saturation of carbon dioxide. So you can see that the graph is shifted towards the right. The green line presents the highest concentration or the highest partial pressure of carbon dioxide. Now, if the graph is shifted in here, this means that we are having lower affinity for the hemoglobin to oxygen. That's why it's shifted in here to the right, and that's why it's taking us more time to load the hemoglobin. Now, we have multiple things. It's because higher concentration of carbon dioxide, which will lead to lower pH. By the way, lowering the pH is not always caused by high concentration of carbon dioxide. There is other reasons, but the one that you need for your discussion is the effect of carbon dioxide. And then having higher temperature, vice versa, shifting it to the left. And by the way, if you're having high concentration of oxygen, the graph will be shifted to the right, as in cases of living in high altitude or a medical condition called polycythemia. We'll get into this, inshallah, later on. So the effect, the Bohr effect, is talking about the high concentration of partial pressure of carbon dioxide. Increasing the levels will reduce the hemoglobin affinity to the oxygen. So why it's very important to discuss this, because without this phenomena, we won't be able to unload the oxygen to the cells. Do you remember when I said that we need to load the oxygen into the red blood cells, specifically into the hemoglobin within the lungs, right? So we must have higher affinity, and this is what happens in the lung. But what will cause the same red blood cells with the same hemoglobin, what will cause it to unload the hemoglobin with oxygen into the cells? It's because of this effect. Of course, there's an enzyme. You don't need to know about this enzyme and its function. So this is extremely important. Now, how I'm going to be asked about this question. The Bohr effect only came in January 2024 as a complete question. However, the style of questions, we finished already the core discussion of today's lesson. The style of questions about the oxygen hemoglobin dissociation curve will be the following. How certain living organisms will adapt to live in this environment that they are living. We'll get into them. And they can get oxygen from it. For example, the first example that we will discuss is going to be called the fetal hemoglobin. You already know that the fetus is living inside of the mother uterus, right? So they cannot breathe. Then how they can get their oxygen? Through the placenta and carbon dioxide. They get the nutrients, the carbon dioxide through the placenta, and they get rid of the waste products through the placenta as well. Except, of course, for the urine, it's going to be flowing in the amniotic fluid. Now, the carbon dioxide and oxygen, what I'm trying to say is that they will go over the umbilical arteries, umbilical veins, back into the placenta. The fetal hemoglobin, do you remember when I said that the hemoglobin consists of 2 alpha and 2 beta? The fetal hemoglobin, however, contains 2 alpha and 2 gamma. Why I'm highlighting the presence of gamma? Imagine this. If the hemoglobin of the fetus is identical to the hemoglobin of the mother, which is 2 beta, 2 alpha, do you think what will drive this oxygen inside of the mother's blood, inside of the mother's hemoglobin, inside their blood cells, inside the blood? What will drive it to move back into the fetal hemoglobin? If we don't have higher affinity in the fetal hemoglobin, it won't be able to move. Diffusion by itself and having higher concentration by itself, it's not enough. So in order for the fetal hemoglobin to extract the oxygen from the mother's hemoglobin, it must have higher affinity. And it won't be able to have higher affinity if they have the same structure. So that's why they're having a different structure. So since it's do you remember when I said presence of carbon dioxide will lower the pH, causing the medium to be acidic, causing the blood to be acidic. So we are reducing the affinity of oxygen to hemoglobin. We are unloading the hemoglobin with oxygen, so oxygen can diffuse into the cells. Here, we're having higher affinity, right? And since we're having higher affinity, we can load the hemoglobin much faster. So it's not going to shift to the right. In this case, it will shift to the left, right? And why it's going to shift it to the left? So the shape of the graph, this is extremely important to know, whether the shape of the graph will be shifted to the right or the left. The shape of the graph in here will be shifted into the left because we have higher affinity, right? So more oxygen can go, so hemoglobin can be loaded with oxygen faster. And this is very necessary for the fetus because they cannot obtain their own oxygen. This is for the fetus, the first example. The second example will be the following. If I'm having animals that lives in high altitude, let me show you this. Not this one. Here it is. So high oxygen affinity in organisms that lives in high altitude. We have the llamas and yaks. And by the way, both of these were tested in the exam. You're not going to see a question about them. For example, you'll see elephants. You're not going to see these concepts within your book. You're not going to see them in the specification. But this is an application of knowledge. So this is a sort of assessment. Usually we call it assessment objective. The start of questions in these were assessment objective 2A and B. Now, you'll be getting questions about the llamas and yaks, and we didn't learn about these. Yes, but see the shape of the graph. Where do these animals live? They live in an area where we're having a high altitude, and you already know that in high altitudes, we're having what? We're having less oxygen. So in order for them to adapt into this environment and living in an environment where we have less oxygen, they must be able to have a higher affinity of hemoglobin to oxygen so that they can live and adapt within this environment. So at high altitudes, the oxygen levels are low. There was a question, if you are taking Unit 1 and you already finished your IGCSEs, people who are having, for example, sickle cell disease or having any issue with their red blood cells, whenever they are feeling into high altitude, living at high altitude, or if they are going into high altitudes, they will feel tired and exhausted. Why? Because there is less oxygen and they already have a condition where their red blood cells cannot carry enough oxygen. So this is something to link it. However, yes, in high altitudes, we have less oxygen levels. So that's why they need to have higher affinity. The second example is going to be for the deep diving mammals. This is not as high yield as the third one, but it's the same concept. Like whales, they live in lower oxygen in areas where there's less oxygen, so they must have a higher affinity. Human fetal hemoglobin, because they need to extract it from the mother. Now, let me tell you something. In all of these, since we are having higher affinity, right? Let's clear this out. So the shape of the curve will be shifted where? To the right or the left? I'm having higher affinity, so it should be shifted to the right. I'm sorry, to the left. But for different reasons. For the yaks and llamas, they are living in high altitude, so it will be shifted to the left. And the deep diving mammals, they have less oxygen because they are living deep in the water, so it will be shifted to the left. For the fetal hemoglobin, it will be shifted to the left, higher affinity, but for different reasons. The main reason is to make them adapt to the environment they are living at. Now, the low oxygen affinity in active species. Now, these species, we are having now the opposite. If I'm having an organism that has a very high metabolic rate, it means that they have more reactions. Now, if you think about it, we're not talking about reactions in the lungs. Of course, lung cells, they do need, by the way, oxygen in order for them to do their own respiration. But when we talk about the tissues or cells, more reaction. So I need more oxygen, but where? Into the cells, right? If I need more oxygen into the cells, do you think that I want to have higher oxygen to hemoglobin affinity? No. I would like to have lower affinity. Why I need to have lower affinity? Because I need to unload this hemoglobin. And unloading of the hemoglobin, this will cause the oxygen to diffuse back into the cells, so they can have or they can maintain this high metabolic rate. And this happens in smaller organisms like mice. They are extremely active, so that's why they need more oxygen in the cells. Therefore, they have lower affinity. Now, if I'm going to ask you a question, you can pause the video. What do you think about the shape of the graph in this? The shape of the graph or the curve in these animals is going to be shifted where? Left to right? Your answer is correct if you said it will be shifted to the right. So this is an application of the concept that we discussed. The fish and adaptation, I'm just adding like examples, even if they're not in the book, even if they're not in the specification, but you get the concept. Let me tell you about, I already put a template how you can answer this question. Number one, I can say that 95% of the questions related to this topic, they will give you a graph. And whenever they are going to ask you about the graph, number one, you have to look at the shape of the graph. Usually they are going to give you two lines. The first line, let's say, it's the adult or the human. And then they will give you different graphs, whether it was right or left, depending on what is the discussion is about. So the first thing that I need to notice is that, for example, they gave me the ox, right? So the shape of the graph for the ox is going to be like this. Or the fetus. The same thing for the fetal hemoglobin. So the first thing that I must know is comparing the shape of the graph. Is it shifted to the right or shifted to the left? This is the first thing. And then I must give an explanation. Analyze whether this shift indicates a higher affinity of hemoglobin to oxygen or lower at higher partial pressures. And then based on this, I must talk about these adaptations in order for the organisms to live. Let's go through an example in order for you to understand what I'm talking about. So this is a past paper question. So as altitude high above the sea level increases, the partial pressure of oxygen in the air decreases. So at high altitudes, we have less oxygen. Llamas are mammals that are adapted to living at high altitudes. The graph shows the oxygen hemoglobin or the oxygen dissociation curve for the llama hemoglobin and for the human hemoglobin. Here you can see the percentage of saturation. Of course, why the reason we are having this sigmoid shape? You must link it to the cooperative binding of oxygen. Why it's here, it's gonna be slow, and then why it's gonna flatten in here. And here we're having the partial pressure of oxygen. So explain the differences between the dissociation curve, use the information in the graph. Number one, if I'm gonna go over this question, I will talk about, as we said in the previous template, the shape of the graph or the curve will be shifted to the left. And the reason for this, because llamas, they have what? Despite they are living in areas or in an environment where we have less oxygen, but they have what? They have higher affinity. So the dissociation curve for the llama is left to that of the human. Therefore, the llama has higher affinity because it can saturate or it can load the hemoglobin with oxygen much faster. So these are the two marks. Now the llama hemoglobin will be fully saturated with oxygen at lower partial pressure. This is the reason, the whole idea for this adaptation. And then you must give a reason. So always, let me tell you about something. The point number 1, 2, and 3, it's always fixed. It's always what you're going to write. Now, number 1, you're just going to change of course, the organism mentioned, left or right. And number 2, you're going to mention whether it has higher or lower affinity for the oxygen, whether it's going to be loaded or unloaded. Why you're saying loaded or unloaded? Because if we're talking about mice, it's going to be shifted to the right, or the earthworms. It's going to has less affinity for oxygen. That's why it needs to unload the oxygen to the respiring cells, because oxygen is needed for aerobic respiration. Now, and then you're going to mention the why we need to have all of this kind of adaptation. Is necessary as there is less oxygen available in the atmosphere at high altitudes. So this is, in summary, the lesson about the oxygen hemoglobin dissociation curve. Let me give you an advice. Whenever you want to study a lesson, go immediately into practicing the topic questions related to this question. Go over the past papers, scan them. I'll provide you with the full PDF for all of the questions regarding this lesson, and it will be following every single lesson that we cover. Go over the questions, try to understand them, and create a template that you can write if I'm being asked about this sort of concept. So make sure that whenever the keywords, whatever the keywords that you are writing, these keywords will make you get marks. Because sometimes students, they will write this right concept, but it's because of the way they word their answer, they are not getting the marks. So let's go over the second question. The graph shows the oxygen dissociation curves of adult and fetal hemoglobin. As you can see in here again, the fetal hemoglobin is what? Number 1, the fetal hemoglobin lies where? Left or right of the adult hemoglobin? Left. And why is that? Because the fetal hemoglobin has higher affinity to oxygen at lower partial pressure of oxygen. Even if we have less oxygen, it can extract it from the mother's blood. So it can be fully saturated with oxygen at lower partial pressure of oxygen. This is mark number 3. And then why all of this adaptation is important is because it's necessary for the fetus survival. So let's see how many marks within the wordings that I said. So oxygen dissociation curve for maternal adults is shifted to the right of the fetal hemoglobin. You can say that it's shifted to the left. It's accepted. Now, because oxygen needs to diffuse from the maternal blood into the fetal blood, and therefore the fetal hemoglobin needs to have a higher affinity. So these are the 2 marks for this question compared to the 4 marks. But miss, how I'm gonna write? What do I need to write? If I'm gonna give you an advice, always go with the template for the 4 markers. And try to write it down fast, because you only have 2 minutes for this question. If it was 2 marks, this is gonna be 4 minutes. So try to write the full template for the answer. So if I'm writing this, I'll be getting the marks whether it was a 4, 3, or 2 markers. Now let's get into this question. So one rule of hemoglobin is to transport oxygen, and here we're having the graph. What is meant by the term partial pressure, and we talked about why we're having a sigmoid shape. We already went over this. Red blood cells transport oxygen. This was, I think, January 2024. Red blood cells transport oxygen to the cells of the body. Read through the following description of how oxygen is transported from the lungs to the respiring cells. Complete the description by writing the most appropriate words. Oxygen binds to where? Into hemoglobin, right? Inside the red blood cells, or erythrocytes. Binds to this molecule in the lungs, where the partial pressure of oxygen. Now let me tell you about something. The partial pressure of oxygen, it's highest where? In the lungs, right? So oxygen binds to this molecule in the lungs, where the partial pressure of oxygen is highest, right? Compared with elsewhere in the body. The oxygenated blood returns to the heart through vessels called the pulmonary vein. We didn't get into this discussion. This will be in topic 1B. And it's pumped through the body by this part of the heart. We know that the structure of the heart that pumps the oxygenated blood is called the left ventricle. In the tissues, the ability of this molecule to bind to oxygen is affected by the higher concentration of what? Of carbon dioxide. What do we call this phenomena? We can call it the curve shift. We can call it the Bohr effect. So we call this the Bohr effect. Let's check our mark scheme to see. So we talked about hemoglobin. It's having high pulmonary veins, left ventricles. This is not related to our discussion for today. And then we have the Bohr shifter effect. You know that what's in between brackets, it's optional to write. It's not for, if you wrote down Bohr, it's going to be correct. So this concludes our discussion about the oxygen hemoglobin dissociation curve. And as I said, going over the key concepts of today's lesson, for example, if you go over the book, you will see the reaction of carbon dioxide within the water and forming hydrogen carbonate. You don't need to know any of this. I am guaranteeing that there is no single past paper question related to this. Now, they are mentioning it, and it's just mentioned in here for the sake of completeness, but whenever you are dealing with a lesson, please weight this lesson, as this lesson is important, and what are the important concepts within this lesson. It's not going into the A level. It's not about having more information. It's about how you can use this information and how you can write them within the past papers. And it's about the skills that you have to develop as you're solving the questions. So yeah, this concludes our discussion for today. And if you're having any questions, please, you can write them down. I will make sure to answer these questions, and I'll provide you with the full PDF that includes all of the notes. And within the course, all of the lessons will be provided with the notes plus their topic questions. And then at the end of each chapter, you should have an exam that looks like a past paper, and you can practice it. And after solving each exam, you will see an analysis for your performance within the weaknesses that you need to work on. After each topic, we will have an exam. It's going to be way longer. And after this exam, I will make sure to write down the performance analysis, and I will give you what are your strengths, what are the topics that you need to work on, what is your estimated score according to this. So thank you for your attendance. And if you have any questions, again, you can ask them, and I will make sure to answer them.